Hello there guys, Saurav Jha back with another topic which is estimation of lipase enzyme. It's enzyme which is responsible for breakdown of fats and which is produced by many organs including pancreas. Okay, so pancreas are main responsible organs which are producing the lipase in our body. Okay, so the lipase, okay, how it acts? It actually attacks 1,3 alpha position 1,3 alpha position in the fats and we can say lipids. Okay, so by attacking on this position it causes the hydrolysis, therefore thereafter break the bond in between the fatty acids to convert them in the simpler fatty acid molecules. Let's get into the topic. First we will discuss some of the main points and some of the roots of the lipase enzyme, from where it comes and why we are gonna estimate it. Okay, so let's get into the topic. The triacylglycerol hydrolase is the another name of lipase. Triacylglycerol hydrolase. Triacylglycerol, which is nothing but a fatty acid, other name of lipid. Okay, so let's get into the next line. Lipase is a glycoprotein. It is the enzyme, and from 12th onwards, from the high school onwards, we know one thing, that the glycoprotein which is written here, it means we know one thing, that all enzymes are protein. Okay, so all enzymes are protein. Lipase is also an enzyme, so it is also a protein, but a special type of protein which is known as glycoprotein. So here we have, it is glycoprotein in nature and have a molecular weight of 48,000, which are international standard unit. Okay, so fully catalytic activity of lipase is present in the presence of bile salt and cofactor called colipase, which means the fully active lipase you can see in the presence of the bile salt, where the bile salt, okay, so let's understand it little deeper. Here we have esophagus and here we have stomach. Stomach is little pathetically drawn, but here we have stomach and here we have duodenum. Okay, so we eat fats came in the stomach, and in stomach we have HCl and some other enzymes also, but the fats are not digested here because pancreas is present here and pancreas will release the lipase enzyme. Okay, simultaneously here we have gallbladder, and gallbladder have bile salts in it, and gallbladder is gonna release bile salts in here in the C-shaped duodenum. Okay, when the bile salt is released in here, sodium glycocholate, sodium taurocholate, these are some of the bile salts, and the bile salts are gonna act as an emulsifying agent for the lipids and fats, and the emulsifying agent will emulsify and make a chylomicron like structure. No, not chylomicron. It makes like this structure, like this. Okay, it's not chylomicron. Chylomicron is at the absorbed it forms when the absorption of lipids actually takes place, but it is a micelle formation. The micelle is formed due to the action of the bile salts, and the bile salts forms this, and from thereafter the pancreatic lipase enters here and then digest the fats. So we know that the full catalytic activity of lipase is present in the presence of bile salt and one cofactor which is called colipase. Okay, most of the lipase present in the serum are produced by the pancreas, which I've told you initially in the beginning of the video already. that most of the pancreas, most of the lipase is produced in the pancreas. Some other organs although release the lipase, but in the very small amount, like the lingual lipase. The lingua means tongue, and tongue releases the lingual lipase. Okay, so tongue releases the lingual lipase, and gastric mucosa also releases gastric lipase. But you will say that, sir, we are not getting one point, that if the fully catalytic activity of lipase is only in the presence of bile salt, then how even the lingual lipase is digesting the fats? So consider one point, that they will not digest the fat fully, they will only digest cooked fat. Okay, they will only digest cooked fat which are already present in some sort of simpler form, which are not in the complex form. The complex fats are acted like this. The complex fats are emulsified with the help of sodium glycocholate, sodium taurocholate, like bile salts, and thereafter the pancreatic lipase acts on the fat, then it gets digested. So thereafter we have clinical significance. So the clinical significance, why are we actually estimating the lipase in the serum? Okay, so if the lipase is increased, then there can be acute pancreatitis symptom. Itis means the inflammation, the pancreas means the pancreas gland, and it means pancreas inflammation of pancreas, which is acute, and the increased activity of the lipase can be seen as the acute pancreatitis. There are other few diseases which can also be correlated with the increase in the levels of lipase. So specifically, if we talk about acute pancreatitis, then we have three cases. The increased activity within 4 to 8 hours is noticed, then the peak comes at 24 hours, within 24 hours, and it goes down with the time from 8 to 14 days. Thereafter we have some estimating methods by which we are going to estimate the lipase enzyme. Titrimetric method, fluorometric method, turbidimetric method, which will have some titration stuff, and fluorometric method which will have some other technical stuff. Turbidimetric method which will include essentially an acid which will react with the fats and create turbidity in the test tube, and which will be seen against the certain color of background, that kind of thing. And spectrophotometric, this is bit advanced, and immunometric enzymatic colored method. Okay, these are some of the methods by which lipase can be estimated. In the upcoming slide, we are gonna looking like we have a one method that will generally deal with a simpler form of test, which is also turbidimetric method. Okay, so let's get into the method straight